Okay, the electrical activity of axons. Okay, now axons is the fiber going away from the cell body in anterograde conduction. So this is really important. You're always going to remember this because we talk about this a lot. The resting membrane potential of the RMP, very negative. And this is negative on the inside, right? Inside the cell is minus 70 millivolts. And the sodium potassium pumps are really ATP pumps or ATPase pumps because sodium is going to be going against its gradient, going outward, two of them or three of them, and potassium is going in, and there's two of them against its gradient. So again, permeability of the membrane is important. And sometimes it's too jumpy. Sometimes it's too permeable to sodium, and you get an inappropriate depolarization or threshold action potential. So I think you know this. Potassium, that cation, is in high concentration inside the cell. Sodium is outside the cell. This all should sound familiar. Altering the membrane potential. So neurons, and we really didn't talk about muscles yet. So muscles and neurons both have excitable membranes. Not every cell can do this. Not every cell works on this electrochemical gradient. We discussed all of the transport mechanisms in the cell membrane. But this is the real excitable cells, and we're going to talk mostly about neurons. Muscle cells can't depolarize on themselves unless you're a cardiac muscle. That's the magic thing about the cardiac muscle. It has autorhythmicity, where it could start its own action potential. Crazy, right? So excitability, I like that better than irritability. Sometimes it is irritable when there's too much permeation of sodium inside the cell, when it's inappropriate. So I like excitability to get a nice action potential out of this. And along the axon, once you change that permeability with an impulse, it continues from segment to segment on the axon, along the axon, to the axon terminal. So again, that electrochemical gradient with sodium on the outside, which is positive, negative inside. The potassium has very little effect on the negativity, on that minus 70 millivolts. It only helps when it leaves the cell to repolarize. So that's a really important fact. Okay. So opposite charges and currents coming in along the axon fiber. And I call the axon a nerve fiber. It's still a neuron, though. It's not a nerve, right? It's not a nerve. It's a fiber. It's a nerve fiber, but it's not a nerve, because a nerve is a bundle of sensory, motor, and maybe autonomic neurons. Right? So now we want to change the membrane potential. And I got to be honest, I really don't know how this happens in somatic muscle contraction. The stimulus starts in your gray matter of your cerebral cortex, starts as stimulus and depolarizes a somatic motor neuron. So that neuron is polarized. Polarized means it's positive on the outside, negative on the inside. And I'm not going through that sorority house thing again, because you know it all already. When you have polarization of a membrane. So now, if the inside of the membrane becomes more positive, and that happens, the best way to do that is to bring sodium into the cell after a stimulus. And the sodium channels will open. And then it's going to become depolarized. Now, it can't stay depolarized very long. I mean, you're going to hear about graded potentials versus action potentials. But an action potential is quick. It depolarized, the membrane is depolarized from negative to more positive over a threshold. And then an action potential is all or nothing. Once it reaches that level of depolarization, it's going to continue down that axon. And it's going to repolarize. It doesn't stay in a positive gradient. The action potential has to stop. And it always does. Okay? Or else you're going to have a problem with whatever target organ you're going towards. So going back from an action potential to repolarization is very important. because that goes back to the resting membrane potential. So remember, depolarization is sodium influx into the cell. Then you'll have an action potential. And then the action potential is all or nothing, but has to repolarize. And in repolarization, the potassium comes out of the cell, and that's efflux. And of course, the sodium channels will close upon repolarization, but that starts at the peak of action potential. So sometimes you can get excess potassium out of the cell, which would make the inside of the cell more negative on a resting membrane potential. Or if you have chloride, which comes in the cell, so this is, let's say, hyperpolarization, right? This is where I'm going to. Hyperpolarization can be excess potassium efflux or chloride influx, because chloride is negative. So if that comes in, it's going to hyperpolarize the membrane. Now, if the membrane is hyperpolarized, it's going to be harder to get an action potential. And I believe we talked about this. So certain chemicals like neurotransmitters can be inhibitory or excitatory. So remember this, all excitatory neurotransmitters, when they bind to the receptors, are going to cause depolarization, sodium coming into the cell, action potential. That's exciting. Okay, sometimes that's inappropriate though. So some postsynaptic membranes have receptors for neurotransmitters that are inhibitory. And if that inhibitory neurotransmitter binds to the postsynaptic membrane, it's not going to allow sodium into the cell. It's going to remain closed, those sodium-gated, voltage-gated sodium channels. But it may open up a chloride channel, which will come into the cell, because chloride is more abundant in the extracellular fluid, not inside the neuron or the muscle cell. And also, potassium can leave the cell. And that, again, is trying to maintain the resting membrane potential, but it also can cause a hyperpolarization. So you're going to see that to get an action potential, you have to have depolarization to about minus 55 millivolts. And that's the threshold. So if enough sodium comes in to go from, and remember the RMP is minus 70 millivolts. So if you have enough depolarization to lower that negativity or increase the positivity, and it gets less negative, minus 55, that's a threshold for an action potential. Then you could reach an action potential, and off it goes, down the axon from segment to segment. Sometimes saltatory conduction, right, over the Schwann cells or myelinated cells in the PNS and the oligodendrocyte myelin in the CNS. So an action potential could peak out in a neuron, I'm going to say plus 30, sorry, it's very positive. But it can't stay like that, so it has to repolarize, and repolarization is back to the resting membrane potential, which is minus 70 millivolts. Now, if this happens, or if it's or this especially, like if it's overridden, then you can get hyperpolarization. And let's say for a neuron, a hyperpolarized membrane could be about minus 90 millivolts or more. So that's a hyperpolarized membrane because there's too much potassium outside the cell in this case, right? And also, it depends on the neurotransmitter, whether it's going to hyperpolarize to this, then it's much harder to get the action potential, or at least to get the threshold. Right? But minus 70 is a lovely number for a neuron to maintain a resting membrane potential and be ready for a stimulus or a depolarization to cause an action potential when appropriate. And this is going on all the time. So this is just the kind of machine that uses an oscilloscope that just shows you the increase in millivolts. right? So minus 70 millivolts, you get a stimulus, right? Right there, stimulus. My cerebral cortex says, move that pen, all right? And if that goes above minus 55, off it goes into an action potential, right? To minus 30, I'm seeing plus 30, and then it should come back to resting membrane potential. But if it goes below, it's hyperpolarized, or even further down at minus 90. So this is a voltometer that basically reads the inside of the cell. So this amplitude right here, or voltage, I should say, is the inside of the cell recording, and shows you on an oscillator, that little hill that I showed you. You could spend hours with those things. It's crazy. So here's depolarization, a lot of repetition, right? Changes in the membrane potential. So sodium usually, because this is massive amounts of sodium in your interstitial fluid in that compartment. Hyperpolarization, when potassium leaves or chloride enters. No surprise. So depolarization of a postsynaptic membrane is going to be excitatory, especially if it reaches that minus 55 threshold. The lights go on, but it's all or nothing, right? An action potential is all or nothing. It's like flushing a toilet. It's not coming back, my friends. Hyperpolarization, again, on the postsynaptic membrane neuron, is inhibitory. All makes sense. So you have a couple of different types of channels. Voltage gated are, of course, what we talk about most, but you have leak channels or leakage channels. And sodium has these too. It's not just potassium, but it makes more sense to have more leakage channels for potassium. So where's potassium most of the time? It's inside the cell. So it has a tendency to leak outside the cell, downhill with its gradient, not against its gradient, with its gradient. And that really helps maintain the resting membrane potential sometimes. It stops like an inappropriate depolarization, because it has to come out to repolarize. So you have to repolarize it back to the resting membrane potential. And voltage gated channels, of course, are important after the action potential. When the sodium gates close, the voltage gated potassium channels open, and they have to close once that RMP, and that's the membrane potential we're talking about, RMP. So potassium is more important, and there's more leakage channels when it comes to maintaining the resting membrane potential. Right, okay. And they're less, sodium now is less permeable. Now that would be completely pathological. If you had, imagine, especially in the heart or in your brain, where you have too much sodium entering the cell, it's a constant depolarization. You're going to get neurotoxicity if you have too much sodium coming into the cell with too much depolarization, too much action potential. And the heart is the best example, because when sodium comes in, it causes your heart to be a little jumpy, and you could wind up with things like arrhythmias or tachycardia at rest, of course, if your sodium channels are a little bit too open. Inappropriately open. And the same thing you're going to see with calcium. But calcium is not so much about this depolarization, repolarization. That's why we're not really talking about it. Calcium is more about the strength of a muscle contraction or the release of a neurotransmitter, which you're going to see. So homeostatic calcium is very important for muscle contraction, whether it be cardiac, smooth, or skeletal. Calcium is really important for the release of the neurotransmitters from the synaptic end bulbs or synaptic terminals or axon terminals, whatever you want to call those, the end of the axon. So again, the voltage gated channels, if it reaches this threshold, right? And that's what we're going to memorize. You'll never forget that. So if, let's say it doesn't reach the threshold, right? Say it doesn't reach the threshold. Sometimes you get depolarization that's always going to be sub-threshold, right? Sub-threshold. Think about this, right? And we probably talked about this already. I'm not sure, because I haven't seen you guys in about a year, right? That's the last time I saw you guys, girls, doctors, lawyers, whatever you are. right? So think about this. Like Chris, this morning, did you put underwear on?
Hold on, let me check. Yeah, I
Yeah,
did.
see, he had to check. He had to check, right? Because he's not feeling his underwear all day, or his earring, or my necklace, or a t-shirt, or a bra, whatever we have that it's going to stimulate a receptor, right? And you're going to feel it, but you don't want to get above threshold to be reminded every 5 seconds. That would be hyper stimulation, right? Does that make sense? Right. So sometimes you can have a depolarization that goes on all day, that's sub threshold, even though it's causing a slight bit of depolarization. Like here's a question, Smarty Pants. If the depolarization goes to minus 60, are you going to get an action potential? No, because that's sub threshold, right? So you're still having depolarization. You're messing with the resting membrane potential, but that's okay, depending on the receptors. Receptors for touch, like if you're wearing a necklace or Chris's underwear, whatever we're wearing, it'll go sub threshold until so you're not constantly getting that bombardment to your cerebral cortexes where you cognate something like that. So the point is the um, the sub threshold potentials, and they could be inhibitory too. They could be inhibitory at the same time because both of them are not reaching action potentials. They're called graded potentials. So whenever I talk about graded potentials, hopefully you won't think about Chris's underwear, but you'll think about sub-threshold stimuli, right? And of course, they could be inhibitory. And it depends on what's going on in the postsynaptic membrane. Like what are the receptors there? Are they receptors for, like I mentioned, the two brain neurotransmitters, I believe I did, right? GABA and glutamate, where GABA is inhibitory and glutamate is excitatory in the brain only, though. It changes in different parts of the nervous system. But in the brain, for the most part, GABA is always going to hyperpolarize the postsynaptic membrane where there are receptors for GABA. Glutamate in the brain is always going to excite the postsynaptic membrane by causing action potential, right? Inhibitory neurotransmitters, when they bind to their postsynaptic receptors, are going to stop an action potential, hyperpolarize the membrane so it can't reach action potential. Does that make sense? Chris, thank you for sharing. We'll move on. So I just want to I just mention, like, why is the threshold so important for action potential, and what happens if you don't reach the threshold? And those are called graded potentials, because that's really confusing when you get to that point, because it's not even really, we didn't really get to the whole action potential story yet. Okay, so if it goes above positive, gets more positive than minus 55, then the voltage gated sodium channels will open along the axon. Right? And then you might get some equilibrium. But either way, an action potential peaks out at plus 30 millivolts. And now it has to repolarize. It can't go through this. Right? And you'll hear what the term refractory means. Refractory, just to give you a heads up, is a period within this action potential pathway where even if you get a strong stimulus, you can't generate another action potential. It's not hyperpolarized. Well, it's part of hyperpolarization, but refractory means you can't get another action potential no matter how strong the stimulus is. And refractory is really important, and really, really important in cardiac muscle. It's a very small period of time, too, very light period of time. Look at this. Here's our old friend, the neuron up here. This looks like a multipolar neuron with your dendrites, and this is your axon. Don't worry about the myelin right here, but I like to see myelin there. So you have a channel that's closed, right? That's closed, impermeable. And then after depolarization, the channels can open. Let's say first, and talking about sodium coming in, right? So these are voltage gated. Now, when it gets to plus 30 millivolts, it's really hard to get another action potential at that point. So basically, the channel is inactivated by another protein. So no more sodium can come in, no matter how strong the stimulus. No more sodium can come in, and that produces a period of refraction or refractory period. So really, all you really have to know is that there is a refractory period within that 0.03 milliseconds of that action potential, where it can't get another one. Kind of important. Okay, and this is going on. At the action potential, plus 30 millivolts, the potassium channels will open, and that's what's giving you the hyperpolarization, excuse me, repolarization, because the voltage-gated potassium channels are now open, and they can repolarize back towards what? RMP. So this is why I say potassium is so important in maintaining that resting membrane potential. What is that number? Minus 70 millivolts. Okay. So I think we got this. And it will come, once you do, here's a key point though on this page. Once you get to the threshold, the membrane and the channels become more permeable to sodium. That's why it's all or nothing, because it opens up the channels along that whole axon. It's not stopping. It's making that depolarization wave, if you like, is going to go all the way down to the axon terminal. Synaptic end bulb is also known as an axon terminal. And hopefully we'll get there soon, because that's a really interesting part of the neuron and the synapse. So positive feedback loop, I guess you could say. There's no stopping it. There's no control. Like flushing the toilet, there's no feedback there, right? Off it goes, or shooting a bullet from a gun or a paintball from a gun, right? So again, this is all depolarization, which ultimately leads to an action potential. I guess if we keep talking about it over and over, it'll make sense. Okay, and then you have the repolarization, which is more of a negative feedback loop. So, and this has to do with refractory. Once you're going towards repolarization, and the potassium is leaving, now the membrane is going back to resting membrane potential. And even if you get another stimulus within this loop, you're still not going to get an action potential until it goes back to resting membrane potential completely. And of course, if it hyperpolarizes, there's no way you're going to get an action potential. So technically, that causes a period of refraction in a regular synapse with an excitatory neurotransmitter. So here's kind of what they show you on that voltometer, right? Where it starts out at 70 millivolts is your resting membrane potential, right? And the stimulus, whatever that stimulus is, like, again, it could be me thinking about writing, it could be Chris putting on his underwear, whatever it is that stimulates a receptor or another neuron. Like, remember, sensory is afferent, coming in from the PNS to the CNS, where motor is more coming from my cerebral cortex down to through the CNS out to the PNS for an effector, like to move a muscle. And that's called efferent. So motor is generally efferent, where sensory is generally more afferent, coming into the CNS. So afferent is into the CNS, and you know that the CNS is the brain spinal cord. And efferent is out of the CNS to an effector, like a skeletal muscle for the somatic nervous system, for a cardiac muscle or smooth muscle, or a gland for the autonomic nervous system. So that's a big deal. So here's the threshold. If you have enough sodium coming in, and again, this is in milliseconds, and as the sodium is coming in, once it hits the threshold, off it goes to the action potential, and it's nonstop, and it'll peak at plus 30. So sodium influx, depolarization. So of course you always write dipole on this side, depolarization. And then now the sodium channels have to close, right? The mechanism of that bottom protein closing the channels or inhibiting any more sodium from coming in, technically. And then the potassium channels open, and potassium is leaving, so that's efflux. Now we're on our way to repolarization. And now it should go back to resting membrane potential. But if it dips below that minus 70 to say minus 90 millivolts, that is hyperpole. So between the top of the action potential and hyperpolarization, that's where refractory happens, where we, even if we get another stimulus, like if we give another stimulus here, you might get double action potential. Even here, you can get another action potential. Even in here, sometimes you can get a relative refractory period. So they're going to hear the difference between relative refractory and absolute. So relative refractory means if you have a massive stimulus, you will be able to get another action potential. But absolute refractory is no way, no how are you getting another action potential within the same period of time, really is the best way to say it. Okay, so again, here's going to different ways to look at this. Again, sodium coming in, dipole, action potential, repolarization. Now here's this, and again, there's time, right? The stimulus, let's say the stimulus actually could happen here. And there's a slight latent period in that beginning of this conduction and depolarization. Of course, look how small, I mean, this is milliseconds, so imagine how little this is, less than 0.4 milliseconds. So peaking out here at the plus 30, it looks the same, and then you have the repolarization. So again, this is possibly a subthreshold loop right here. So it depends on how high this is going. So you're getting depolarization, you're getting sodium into the cell, but it never really reaches the threshold. So right away, potassium channels will open and it'll go back to its RMP, and then once it's at the RMP, of course, sodium and potassium gates closed, and you're back to polarization. After hyperpolarization, now repolarization will overshoot. Like I said, minus 90, but it's the same. Minus 85 against well below minus 70, right? So you have too much potassium leaving the cell. So how do we get that back? We try to use the sodium potassium ATPase pumps. And this is really how we maintain the resting membrane potential. And I say it's ATPase because really these pumps are an enzyme pumping 3 sodium out, right? And 2 potassium in, right? Against their gradient. And so you need energy. So you have to hydrolyze ATP to ADP plus the inorganic phosphate to use the energy to make this pump work. And this is on every one of our neurons and muscle cells. Very important. We spend a lot of our ATP on these pumps, actually. If you really did an inventory of how we use ATP, a lot of it's used just to maintain that resting membrane potential of your nervous system, your neurons. That's how important that is. So now it's action potential, and you should have an idea what we're talking about. So this is all or nothing, all or none law. Flushing the toilet. All right, it'll happen once you reach that minus 55 millivolts. Off it goes, and it always reaches the plus 30 millivolts and peaks out. That's been well documented. Okay, so it doesn't matter how much of a stimulus or how many stimuli you have, it's the same every time. All right, it's the potential of getting another action potential, not making the one action potential more positive, right? That really doesn't happen. That's a really good point your book makes here. So every action potential is basically the same, no matter how strong the stimuli is. And that's a really controlled mechanism when you think about it, and it has to be homeostatic. Again, we're looking at different stimuli, like from weakest to strong, right? But it's the same. This is an action potential. These aren't graded potentials. These are action potentials that reach threshold. So again, remember, sometimes it won't reach the threshold, right? but this is action potential. So they all peak out at the same place. This is an excellent point. Starting at your resting membrane potential of minus 70 millivolts. Again, you can get more frequent action potentials, but it's still different action potentials, right? And again, a stronger stimulus may recruit other neurons within a circuit or a pathway. But all we've been looking at is one neuron stimulating another neuron. And technically it's the same if one neuron stimulates a muscle cell. So now we're talking about multiple neurons within a nerve or within a pathway, like a tract. A tract is a group of neurons together, like motor tracks coming down from the CNS, like from the brain to the spinal cord, then out, or tracks coming in from the peripheral nervous system, right? I think I mentioned two tracks, like the corticospinal tract is a group of neurons running from the cerebral cortex through the spinal column, or spinal cord, sorry, and then out to the muscles, which are the effectors. That's a somatic nerve tract. Now coming in is called spinothalamic. So the input comes in from sensory receptors into the spinal cord and then up to the thalamus, and the thalamus basically relays all sensory afferent information to the cerebral cortex except smell. So that's called the spinothalamic tract. So this is many neurons being recruited for the same purpose, basically, but each neuron itself has the same action potential. It's one single action potential from 1 to 1. So the frequency and the strength of the stimulus only recruits more axons, or I should say more neurons. That's a really important fact right there. Okay, so you could have more spaced out action potentials, right, depending on the stimulus and the amount of stimulus. These are multiple stimuli, right, just looking at that. So again, this is action potential. This isn't graded potential, because sometimes once you have an action potential, it stops right away. But a graded potential, like I was trying to tell you before, is something that can go on for a long period of time. And its distance is very short, where action potential could propagate all the way down to the end of the longest of neurons. Like, did you ever hear of the sciatic nerve? I might have mentioned this too. Sciatic nerve is a lumbar plexus nerve that has many, many axons and many, many neurons. But it's the longest nerve. So the neurons that are in the sciatic nerve, motor and sensory, could be like 3 or a meter long, like 3 feet long. That's pretty long. So again, that is the length of the neurons important, the amount of stimulus, the frequency and the strength of the stimulus, and the duration of the stimulus as well. So that's what this is basically showing us. Now refractory, hopefully I introduced that, because this is a complicated point. All right, action potentials can go on one at a time, right? And when you have refractory, that neuron and that membrane of the neuron, should say, cannot become excited again. It's not possible to have more depolarization. So the absolute refractory is when the sodium channels are completely inactive, right? They could be open, but they're inactive, like you saw in that last picture a few slides ago. So sodium, again, cannot get into the cell, leads to absolute refractory. Refractory is no way, no how are you going to get another action potential, no matter what the stimulus is, how strong or how many at that point. And it's a very short period of time. The relative refractory period is when the potassium channels are still open, like during repolarization. Right? So a very strong stimulus during repolarization can get another action potential. So again, each action potential still remains separate. One action potential is one action potential. So this Relative refractory period means you, with a very strong stimulus, you'll get another action potential. But most of the time you won't. Most of the time you won't. So it would have to be very strong. So the absolute refractory period is pretty much the whole length of the action potential. This should be familiar to you now, right? So you can't get an action potential in this first millisecond, whatever it is, coming down to the resting membrane potential. So after repolarization and hyperpolarization, of course, there's no way you're going to get a action potential, because now, I mean, there is a way. If you have a really strong stimulus here, you can get an action potential. I'm sorry. Okay. So again, hyperpolarized membrane, still minus 85, minus 90, you may get an action potential, but it's still called a relative refractory period, because most of the time you won't unless it's strong enough. So absolute is right during the action potential. It's not going to happen. So you should get used to what this looks like, especially the numbers, which again, we repeated a lot. Hopefully you're good with that. Okay. Again, this is just talking about the impulse being carried along the axon, right? And again, it depends on the resistance within the axon, which could be pathological, and the moving the impulse down the neuron. Remember, if it has myelin, the cable properties of the neuron is going to be very fast and very organized. Without myelin, it's slower, and it's more likely that there could be some type of problem with depolarization, like CNS, especially like interneurons or unmyelinated fibers within the CNS only. Interneurons are usually unmyelinated almost all the time, and they're the most abundant neuron type as opposed to sensory and motor. So interneurons are integrative, short distances. They're not long distances like some of these motor and sensory axons that are myelinated, even within the spinal cord. So it really depends on a few things, which we'll go through. Again, this is the movement along an axon, so you kind of have to picture the axon terminal is here, right? So it'll move from segment to segment, the depolarization, all the way down the neuron and ultimately reach the axon terminal. And it's repolarizing at every segment as well, right? So look at this. Now we're looking at subthreshold. And let's see, what's cable properties. So this is subthreshold depolarization, where it's going to continue, and it can continue for a long time, but it's not reaching the threshold. And this is what's going towards what's called graded potential, right? Graded potential. Looks like they're injecting positive ions there. Subthreshold depolarization, though. Sorry, this keeps getting in our way. Perfect. So when an action potential occurs, the voltage-gated sodium channels open, and it goes down the length of the axon. So, and let's keep it as a one-way direction from the cell body trigger zone, right? The axon hillock, the initial segment, which is the beginning of the axon, will summate actually and kind of find out if it's going to reach threshold to have an action potential. So once the action potential or the depolarization reaches the axon terminal, it could move it across to the next neuron in the synapse, which we'll talk about. The next axon, if you will, the next region of axon in this case. So it goes from region to region and then to the next cell, which will be technically is the dendrites of another cell. So most synapses, and of course the synapse is the gap junction between neurons. So it's usually axodendritic. So an axon is the terminal. So once all of the axon has depolarized to the bottom and repolarizing as it goes, once that depolarization action potential reaches the axon terminal, now it can transmit it to the next cell, which is usually a neuron, sometimes a muscle, because those are the two excitable membranes on the cells that we study. Again, just showing the action potential beginning and moving down towards the end of the axon. So so far everything has been anterograde, right? And I like the picture here. Here's your dendrites. This is where the information comes in this synapse. It's a gap junction to a synaptic cleft. And then this zone right here, between the axon hillock and the initial segment of the axon, is called the trigger zone. I don't even know if books use this term, but I like this term, trigger zone, because it's not really an anatomical part. It's more physiological within this segment between the initial segment of the axon, which is here, and the axon hillock, which is there. So right here, all this incoming information, right, which could be inhibitory or it could be excitatory, it's all going to be added up in a place called the trigger zone. Again, this is kind of like more of a metaphor, but it is in this part of the neuron. So all the information, let's say it's excitatory, inhibitory, depending on the neurotransmitter, right? Excitatory. And this could happen, 10 times, 1,000 times. Inhibitory. So this area summates all that info, and if it summates above threshold, off you go with depolarization along the axon and these are the axon terminals down here. So that'll meet up with another dendrite of a subsequent cell. So we're going to stick with the one directional action potential depolarization towards the end of the axon, which is called the axon terminal. And each segment repolarizes after it depolarizes. So it's a segmental cable of depolarization all the way down to the axon terminal. And the axon itself, once that's finished, is ready for another action potential because it reached its resting membrane potential because each segment repolarizes as it goes down the axon. Now, unmyelinated neuron, right? So it's not saltatory. It doesn't jump from node to node, right? Node of Ranvier, right? It doesn't go over the neural lemma. It goes straight down the axolemma, right? And it's slow because it's not happening saltatory. It's not jumping from each segment. So everything's the same going down, and it goes kind of slower, right? So it's one individual event. And again, these aren't neurons in the peripheral nervous system that are connected to or synapsing with sensory neurons. Like you're going to know right away if you step on a nail, right? Because of myelinated fibers, they basically cause a reflex in the spinal cord, but then also reach your cerebral cortex, which tells you you're stepping on a nail. But in between the neuron that goes to the cerebral cortex or goes to the thalamus in many cases is an interneuron, and the interneurons between the PNS neuron and the cerebral cortex neuron is usually an interneuron. And interneurons are generally unmyelinated, multipolar neurons. They look like motor neurons, but they're unmyelinated. So that's important. And they're called integrative, like the interneuron's called integrative, and usually they're not myelinated. So myelinated, of course, remember Schwann cells in the PNS and oligodendrocytes in the CNS. And they're different. They look different, but they're both producing myelin. Remember, myelin is what makes white matter white because that glycoprotein or glycolipid, is white when you look at it along those nerve tracks. So this improves the speed and the organization, too, of the cable properties of each part of the axon conducting a depolarization action potential along the length of the axon. So at the nodes of Ranvier, and nodes of Ranvier, if you remember, are unmyelinated nodes between the myelin. And you have sodium-potassium channels there, which makes a lot of sense, right? So they kind of skip some axon space or axon longevity to go to the axon, the sodium-potassium channels, which of course increases depolarization and repolarization as you're going down to the axon terminal. So leaping or jumping from node to node speeds everything up, and this is what's called saltatory conduction. So when myelin is present, we have saltatory conduction, which of course is faster. So this kind of shows that, the myelin around, and this looks like a motor neuron, which is multipolar, and it's myelinated. So the cells that myelinate in the PNS, this would be PNS, these are Schwann cells. And in between you have your nodes, or nodes of Ranvier in between. So along this membrane, which is called the neurolemma now, is saltatory conduction. So you have a jumping in between, and the sodium channels, of course, open to depolarize. And it continues. So the axon or the axolemma, of course, carries the charge, or carries the voltage-gated potentials. So without myelin, it's going to be like one movement straight back, where saltatory is depolarizing, then repolarizing, depolarizing, repolarizing, depolarizing, and repolarizing, all the way to the axon terminal. So keep this one direction conduction in mind. And this is some of the reasons. It's always a question, like on a test, just like what increases the speed of an action potential. And usually it's the diameter of the neuron and the amount of myelination, right? So the thickness of the fibers, which it changes in certain places, especially in the PNS. Also, another one is temperature. And of course, that needs to be homeostatic at 37 degrees Celsius. So temperature is also another factor that could speed up or slow down the speed of a conduction along an axon. So this just gives you an example of the speed difference, right? Unmyelinated versus myelinated. This is myelinated with saltatory conduction. Unmyelinated neurons, they conduct what's called continuous conduction. And that's along the axolemma, and this is saltatory with myelinated, jumping from node to node. Okay, just showing some diameters and conduction velocity. Like you could measure this, like they have, like if you go to the doctor and neurologist, they can do conduction velocity tests and compare it to what it should be for your demographic, or sometimes they'll measure side to side. If you have, like, say, a right-sided nerve impingement or a problem with that nerve for whatever reason, it could affect the conduction velocity. So that's kind of practical where that can be used. So looking at the diameter in micrometers, you could see that it's directly proportional, right? The diameter is directly proportional to the conduction velocity in meters per second, believe it or not, right? So where do we find these? Like where are the thinnest ones? Let's see, autonomic fibers, right? Autonomic fibers, which are unmyelinated from sympathetic and parasympathetic. And they're peripheral too. So autonomic are generally thin and most of them unmyelinated, right? Autonomic fibers. When we talk about the autonomic nervous system, you'll see how that's set up. So these are all fast, right? These are all fast. So pain and temperature, again, fast. The sensory touch, right away, like you cognating sensory, right? These are sensory. Motor is faster. Motor is faster. So being able to move a muscle or innervate a gland, but this is really muscle. This is somatic, right? This is somatic. So motor fibers for contraction and movement. Muscle position is more about balance, right? Where you're able to maintain your posture or your joint position, your kinesthetic sense. So, which is really the fastest. Pretty interesting. All right. So it's good to know an example of the neurons that are thicker, or axons, we should say in this case, and the ones that are thinner. Right? So these are more prone to other problems, right? Autonomic reflex problems, which we'll learn about. It really has to do with the synapse itself, which we'll talk about in a minute. Everybody okay? Anybody have any questions at all about any of that? The synapse. See if we can move on here. So this is the connection. And we're talking about neuron to neuron. You could have a synapse between a neuron and a muscle as well, or a gland even, like the adrenal gland. So it is a gap junction, right? And again, PNS, you could have a gland or a muscle. So in the CNS, it's all interneuron to sensory neuron to projection neuron. So it's more likely in the CNS, the central nervous system between the brain and spinal cord, or within the brain. It's it's always with another neuron, except one other place in the hypothalamus, which is posterior pituitary gland, has a neuronal connection. So technically, the hypothalamus is in the CNS, but the pituitary gland is not even a nerve structure, but it does have some nerve innervation right from the hypothalamus. That's a really important area, actually. Okay, so now we have to get this whole presynaptic neuron versus postsynaptic neuron. So the presynaptic neuron at the synapse is basically the membrane of the axon terminal. Right? So remember, axon terminal, sometimes it's called synaptic end bulb, and that belongs to the presynaptic neuron. And at the other side of the synapse, the cleft itself is called the synapse, synaptic cleft. The connection is a synapse. Sometimes I say on the other side of the synapse, but it's really the other side of the synaptic cleft. So the postsynaptic neuron also has a membrane, a postsynaptic neuron membrane. And this, I'm just going to make a blanket statement that most of the time, the postsynaptic neuron, the membrane is a dendrite, or the membrane of the dendrite part of the neuron. It could be a cell body. It could be an axon. But let's keep it simple, where it's an axodendritic synapse. So, but it could also happen in the cell body, where they call it axosomatic, because the cell body is also called soma. Or it could be an axoaxonic. So the axon of the presynaptic neuron can synapse with the axon of the postsynaptic neuron. But axodendritic is the easiest to understand. It makes the most sense, and it helps you learn too, I believe. Right? So the presynaptic neuron can signal the dendrite, the axon, or the cell body. So I would say the axodendritic is the one we'll talk about the most of the time. Okay, and it's one directional. It's easy. If for learning processes, it's much better as an outcome. Right, and synapses can be electrical, which is voltage gated channels, or sometimes there's connexons, which are like channels that go from the presynaptic neuron to the postsynaptic neuron. Chemical is about neurotransmitters being released. So the electrical is more about your ions, or electrolytes, whatever you want to call them. Really important. So it's smooth, with smooth muscle, right? Smooth muscle. Again, smooth muscle is completely involuntary, which is controlled by the autonomic nervous system. So that's an autonomic. Cardiac muscle has its own neurological circuit or conduction system, but the cardiac muscle is controlled by the autonomic nervous system, like to speed up the heart rate, increase the contractions or the strength of contractions, or decrease, depending on sympathetic versus parasympathetic. Same with smooth muscle. Smooth muscle is your blood vessels, the walls of your blood vessels, the walls of your digestive system, respiratory system, reproductive system. Okay? So some neurons of the brain, or between the synapses, could be between the neurons of the brain and between glial cells. All right, remember what glial cells are, right? Glial cells are those supportive cells. They're not neurons, right? The 4 in the CNS, and there's 2 in the PNS that we need to learn, or we learned. And these are gap junctions, and that gap junction is called the synaptic cleft, or just synapse. Cleft is kind of a space, but it's a fluid-filled space. It's not just air. Okay? So there can be connexin proteins, right? And this is about the ions. These are about ions going from one neuron to the next. So again, that includes the phosphorylation or dephosphorylation of those actual proteins. Now remember, proteins are embedded in the membrane. Now some are connexin proteins. I'm not going to talk a lot about these though, to be honest with you, because I like to talk about the neurotransmitters and their protein receptors, because they're attached to channels as well. And we'll see what that means. But these are straight up voltage gated channels, really, connexins between 2 neurons through the synaptic cleft. So they open and close based on electrochemical gradient. So they're more about electrical synapse, not so much chemical when you talk about these connexin proteins. So these are about ions, electric, because the receptors are going to be for the neurotransmitters. So technically, this is kind of like a bridge between, and here's the synaptic cleft in here, and you have your presynaptic, say, membrane and your postsynaptic membrane. Remember the plasma membrane, right? And proteins embed into those membranes. So they're connected, really. That's why they call them connexins, and they're proteins transcribed from DNA. Right? So this is the postsynaptic membrane here. So that's about ion passage. And of course, this is, we didn't learn this yet, but this is an example of cardiac muscle. And these little areas of connexins are called intercalated discs. We'll get to that when we do cardio. So they're protein connexins between your cardiac muscle fibers, which branch. So again, I don't talk about that a lot now in the brain and the nervous system. And I'll mention those again when we do cardio. But I like this part. I like the release of the chemical. So the chemical, the neurotransmitter, is released from the axon terminal of the presynaptic neuron, right? And they call it axon terminal. Sometimes they call them botons, synaptic end bulbs. They have different names. So we don't use this word too much. It's just easier to say axon terminal when you're learning it, because it makes the most sense. Right, so the axon terminal is the end of the axon. So the neurotransmitter is actually stored in vesicles within those axon terminals or botons. Okay, synaptic cleft is very small, as you can imagine, and they're held together. I mean, there is a connection with these adhesion proteins called CAMs. Again, that keeps the integrity of the synapse, but it really doesn't get in the way. And it's just a connection, like any 2 cells are connected, but there is a gap, and there is fluid in that gap. Very, very small, of course. Look at this. Okay, so this is showing the postsynaptic cell. In this case, it's a muscle cell, and they call that the motor end plate, and how small that synaptic cleft is. So all of this is the terminal or axon terminal, and stored in these little vesicles, these little spaceships all in here, right? You're storing a neurotransmitter, in all of those. And of course, you need mitochondria in both, not just the neuron. So this is a neuron. I should say that. This is a neuron, the axon terminal of a neuron. This down here is skeletal muscle. So we're talking about muscle contraction. So anybody remember what the neurotransmitter is for muscle contraction? Somatic. It's also used in the parasympathetic nervous system. Anybody remember? I'll give you a multiple choice. Is it norepinephrine, or is it acetylcholine, or is it serotonin?
Acetylcholine.
Yeah, you got that right. It's always acetylcholine. And acetylcholine is always excitatory to the muscle membrane or neural end plate, as they call it sometimes, or sarcolemma. So sarco means muscle. So again, that's muscle. When we talk about cardio, we'll talk about muscle cells more. So this is really important. This is where I like to live. So how is a neurotransmitter released? Right? We know that they're stored all the time in the axon terminal in vesicles. Only one S there. Right? So what has to happen? Let me just go through this and I'll ask you. So number one, when that action potential reaches the axon terminal, voltage-gated calcium channels open. That's a key. This is why calcium is so important in nerve conduction. So calcium enters the cell, enters the axon terminal, and there's a chemical reaction as the calcium binds or fuses with the vesicles that contain the neurotransmitter. It begins this active transport mechanism called exocytosis. So the neurotransmitter, like acetylcholine in the page before, acetylcholine is released into the synaptic cleft via exocytosis, right? So again, a greater frequency of action potentials, like if there's another action potential that follows the one that reached it to begin with, you're going to get multiple or excess release appropriately of the neurotransmitter. So in this case, the case of the page before, it is acetylcholine, but it could be any actual neurotransmitter, right? So let's think about this. So how does this all start? Let's say, now this is muscle contraction, which I showed you on the picture before. Think about this. Like if I want to write with my pen, right? Right, correct. Where does that signal start? Anybody take a stab at this? Because I don't know. I mean, I know where it starts, but I don't know exactly how. So I decide, this will help you. I decide to write with my pen. Where does that signal actually start?
The CNS.
Yeah, for sure. In the brain, and more specifically in the cerebral cortex, the outer gray matter of your motor center, your somatic motor center, because contracting skeletal muscles is somatic, not autonomic. And skeletal muscle is voluntary. So the voluntary part is me thinking about moving my pen. And I really don't know how that happens, where that ghost in the machine comes from and starts that depolarization, but it happens in my frontal lobe. If I want to, I'm a right-handed person. So my left primary motor area of my frontal lobe starts an action potential of an upper motor neuron that goes down to my spinal cord, crosses over, mind you, within my medulla oblongata, and goes to my muscles of my right arm and fingers. So that's, there's a couple of neurons involved. There's an upper motor neuron that goes to the spinal cord, crosses over in between at the medulla oblongata, and then there's a lower motor neuron that comes out of the spinal cord that correlates with my brachial plexus, which is, your neck and upper back, mostly neck, lower neck. And all of those neurons form nerves that go to my arm to write. And I did that all with starting an impulse with my cerebral cortex. So now we get down to the cerebral cortex now, right? I'm sorry, we get down to the motor end plate or the muscle. So what happens next? Now you have to assume that there's action potential that whole way down there, right? There's action potential. So the action potential, all that depolarization has reached my lower motor neuron synaptic end bulbs. Are you with me? So again, it repolarized as it was going down from my cerebral cortex, crossed over in the medulla oblongata, came out C5 to T1 in my cervical nerve roots, lower motor neuron, axon terminals in the muscles of my arm. So once the action potential reaches that synaptic terminals of those neurons, which are multi-neurons and nerves, right? Because now I'm organizing all different muscles and recruiting different muscles to write with. So now what happens next? What has to happen? This is really a key part, if you can remember. What has to happen next? Like what voltage gated channels have to open when that action potential reaches the axon terminal? Before it even goes, before the neurotransmitter is even released. This is where we really have to remember. So once that action potential depolarization reaches my synaptic terminals in those neurons, axons, the calcium channels open, the voltage gated calcium channels open. That's extremely important. So now the extracellular calcium comes into the synaptic end bulb and the calcium fuses with the vesicles. What's in the vesicles?
Neurotransmitters.
A neurotransmitter. And in this case, with me writing, that neurotransmitter is going to be what?
Choline?
Acetylcholine, right? Acetylcholine, I like to say. It sounds more studious when I say acetylcholine, but it's acetylcholine, ACH. So that is diffuses out. Well, actually, it leaves the axon terminal via vesicular exocytosis into the synaptic cleft. So now you have your synaptic cleft is flooded with acetylcholine, right? It's flooded with acetylcholine. So that was all the presynaptic neuron. But in this case, we're talking about muscle. So the postsynaptic membrane is a muscle. It's not a neuron. Everybody understand that? Because we're talking about somatic muscle contraction with me writing with my hand, or you doing a certain dance move, or using your diaphragm to vocalize, whatever it is. So what has to happen to the acetylcholine? How does it, this is a really tough question, but think about this. What does the acetylcholine have to do to cause the depolarization of the postsynaptic membrane, whether it be a muscle or another neuron? And it's excitatory. This is excitatory. It's going to cause an action potential if you get enough depolarization. So what is acetylcholine? It's a chemical, right? It's not going to go into the postsynaptic membrane and make a change. It's not going to go in there. So what does a neurotransmitter have to do in order to make a change on the postsynaptic membrane? Anybody have a clue or guess? It's a tough question.
I guess stimulating the ion in the post.
Indirectly, yeah, that's a good stab. So here's a very important situation here. Now, acetylcholine it's not a lipid, right? It's not a protein either, but it's a small molecule, relatively small molecule that can't just come into the membrane, postsynaptic membrane, and make a change. And the change is, like Chris said, causing a voltage gated ion influx of sodium. That's depolarization. So something like acetylcholine or even norepinephrine or dopamine has to bind to a receptor. So acetylcholine flooding the synaptic cleft is useless unless you have the receptor on the postsynaptic membrane specific for acetylcholine. So once acetylcholine binds to its receptor, acetylcholine never enters the cell. But what acetylcholine does, and acetylcholine is a ligand. The receptor is the receptor for that ligand. In this case, it's acetylcholine. So once that acetylcholine, the ligand, binds to its receptor, now that opens up sodium channels and potassium channels, technically. So that causes that depolarization secondary to the neurotransmitter binding to the receptor. So you need a receptor, specific receptor on that postsynaptic membrane, right? Makes sense. Now that acetylcholine in muscle contraction is always excitatory, always, always, always in somatic muscle contraction. But sometimes we want to inhibit a muscle contraction. So usually an inhibitory neurotransmitter for muscle, especially in the CNS, like in the spinal cord, when it's trying to coordinate movements and coordinate reflexes, a neurotransmitter, actually it's a amino acid called glycine. Glycine is inhibitory, but it still needs a receptor. So if glycine is released to the postsynaptic membrane of a muscle, we'll say, glycine is going to inhibit an action potential and open up chloride channels, not sodium. And what does chloride do to a membrane? Does it depolarize it or does it hyperpolarize it? Chloride, when it comes into the cell. So think of this. Chloride is a negative ion. Sodium is positive. Picture the postsynaptic membrane being in its resting state, resting membrane potential.
Hyperpolarized.
Yeah, so chloride's going to hyperpolarize. And you knew that, right? Makes sense. So in that case, you're not going to get an action potential. You're going to get inhibition to that specific postsynaptic membrane. Now, in muscle, of course, it's going to inhibit a muscle contraction, which is completely appropriate. Like if I step on a nail, right? Or even less. Say I tap you on the knee, the patellar ligament below your kneecap. If I tap you on that knee, you're going to extend your lower leg, right? Your foot's going to come up if you're sitting, correct? So you're contracting your quadriceps muscle. So that's all excitatory, right? Because if you're exciting the quadricep fibers with acetylcholine, you're going to get action potential. You're going to get contraction, shortening of the muscle, and it's going to do the work of bringing your leg up. But what are the muscles called in the back of your leg? Right? There's muscles in posterior thigh, right? There's your hamstring muscles, right? Your hamstring muscles. Now, if your quadriceps are contracting, you can't contract your hamstrings at the same time. They have to completely relax. So your spinal cord has to send that signal during that reflex to relax your hamstrings on that side that I tapped you on with the hammer. So it's an inhibition, right? So that's not acetylcholine. That's going to be glycine released there from the.
Is that what they call reciprocal inhibition?
Reciprocal inhibition. Reciprocal inhibition. Very good. And it could happen on the other side too. It could, like, if I step on a nail, with my right foot, my left leg's got to hold me up, right? So you have to have the same condition on the other side with contralateral innervation with reciprocal inhibition. So when we do reflexes, we're going to mention that reciprocal inhibition. But the point is now is what has to be released and what type of receptors on the postsynaptic membrane. So which neurotransmitter is released, right? In this circuitry. Make sense? I hope so. Okay, how do I get back here? Let's see what else we have. Okay, calcium and synaptic vessels. And we know calcium enters the cell, it binds to this protein, which is a synaptotagmin, and serves as the sensor for calcium on the vesicle. So again, there's snare proteins docked on the plasma membrane, because vesicles basically have their own membrane. A vesicle is actually like a little spaceship that has its own biphospholipid layer as a membrane, right? So the calcium complex, right, synaptotagmin complex displaces those proteins, and now the vesicle can fuse with the membrane, the outer membrane, right, the postsynaptic synaptic terminal membrane, and then it can release the neurotransmitter through exocytosis, and that's active transport. So basically, this complex displaces the snares, those proteins. It's all about proteins, right? So that has to work pretty easily. So not too many things go wrong with that actual process of calcium fusing and then releasing the neurotransmitter through exocytosis. So that's pretty straightforward reaction. The problem is this. The problem is the receptors on the postsynaptic membrane have some integrity. They have to be there and they have to be usable. And if they're not there, or they've been destroyed maybe by some type of autoimmune condition, say, because they're proteins, right? The acetylcholine or the dopamine or the serotonin or the norepinephrine is not going to work. It's not going to do the job it's supposed to do, whether it's excite or inhibit the postsynaptic membrane. So releasing the neurotransmitter is not usually the problem. But what could be a problem? What if your body is low in, or your blood is low in calcium? Right? That's going to cause an aberrant nerve conduction. Right? And if that's the problem, it's going to cause an aberrant muscle contraction, because you need calcium for muscle contraction, just as important. So this is introducing a good point about calcium for nerve conduction, clearly what it does right here by the releasing that neurotransmitter via exocytosis by binding to the synaptotagmin and then displacing these snares and allowing exocytosis of the neurotransmitter from the presynaptic membrane into the synaptic cleft. Makes sense, right?
Professor, I have
Go
a
ahead.
question.
Yeah.
So, I mean, it's in the diagram that you have right now. So, are the vesicles docked to the snare proteins before the
Technically not.
calcium gets to them?
Yeah, the complex
Okay.
is on there. Yeah, the complex is already there. Yes, good question. All right, and then the binding of calcium to the complex will displace the snares and allow for, look how this fuses, right, with the membrane, right? So it'll cause the reaction and it'll cause the actual anchoring to the membrane, because the vesicles, again, are just continuous of the membrane. Like the part of the membrane will actually split off and go here, right? So it's not actually showing you the synaptotagmin so much, right? But technically it's on the vesicle. That's a really good question. Understood? Cool. Okay, so here's the steps. Action potential reaches the axon terminals, and then because of that, these channels, which are voltage-gated calcium channels, and calcium comes in from the ECF, and then the binding of the sensor on the protein complex, synaptotagmin, and then the complex fuses with the membrane and allows for release of neurotransmitter. So like in the muscle end plate, you're going to have receptors specific for acetylcholine, right? So the acetylcholine can bind to its receptor and then open up sodium channels for sodium to come in and now cause, and now this could be a neuron, it could be a muscle membrane. So now you're going to get depolarization of the muscle, if it's a muscle, and that has a threshold too for action potential as well. It's a little bit higher than the neuron action potential. And then you'll go through and along the cell, it'll keep going down the length of the muscle fiber, if that's what it is, or the neuron, to the next terminal if it's a neuron. If it's a muscle, the muscle is just going to do its job by shortening, contracting to do work. All right? Clear? Okay, so again, there's diffusion of the neurotransmitter across the synaptic cleft and has to bind to a very specific receptor. So the neurotransmitter itself is called a ligand. Like in endocrine, you know about a ligand-gated or even a ligand-gated channels. So the ligand means there's a chemical that has to bind to it. usually a neurotransmitter, right? And chemically gated or chemically regulated ion channels are called ligand gated ion channels. And for depolarization, you know they're going to be sodium, potassium, right? Mostly sodium coming in and then potassium later coming out on repolarization, because muscles have to repolarize as well, just like neurons. Okay, so let's take a break and then we'll talk about the graded potentials when we come back. Graded potentials. Now, there's two things you need to know about the postsynaptic potentials before we go into this. There's two acronyms here. There's EPSP and IPSP. So EPSP is a excitatory postsynaptic potential, meaning that on the postsynaptic membrane, you have receptors for excitatory neurotransmitters. And inhibitory postsynaptic potential is receptors for inhibitory neurotransmitters. And of course, you have both, but it's the postsynaptic neuron that's either going to be inhibited or excited. So again, sodium, and especially sodium, is more about depolarization, excitation. And calcium, of course, has to do with a positive ion coming in and also the release of the neurotransmitter. But either one, you need calcium to release a neurotransmitter, whether it's excitatory or inhibitory. So the inhibitory postsynaptic potentials are going to open up the potassium channels, which is more efflux of potassium, or chloride channels that are more influx of chloride, which will hyperpolarize the membrane. So basically what I was trying to describe before, now you could have multiple neurons, presynaptic neurons, affecting the dendrites, multiple dendrites of a postsynaptic neuron. So all this information coming in, all the EPSPs, all the IPSPs have to be summated. And if the EPSPs win, you're going to get action potential, meaning that there's more excitation coming into that neuron than there is inhibition. And this goes on all the time. So the graded potentials are all of these EPSPs and IPSPs being summated. And graded potentials are subthreshold until they go above the threshold, and then it's an action potential. So graded potentials are shorter lived, more frequent, and much longer, go on for a longer period of time, I should say, where action potentials are long in length, I should say, and they're all or nothing, so they go very quickly, where graded potentials could last longer. So again, EPSPs, excitatory postsynaptic potentials, and IPSP, inhibitory postsynaptic potential. So as the neuron, the dendrites, or postsynaptic neuron, are receiving the EPSPs, it's going closer to threshold, but it still could be subthreshold. And there could be many neurons affecting that postsynaptic potential. So the IPSPs are hyperpolarizing, and they're moving the membrane potential away from the threshold, right, which is minus 55 millivolts in this case. So again, they could work against each other in a way, but they have to be summated. And they're summated between the initial segment of the axon and the axon hillock in that space I like to call the trigger zone. It's not an anatomical part. Some books use it, some don't. And that summation of the EPSPs and IPSPs will either have an inhibition of an action potential or excitation causing an action potential. So that's the determination if that action potential is going to move along the length of that axon towards the axon terminal. And then to the next neuron, same thing. In the synapse, of course. So great, again, great picture here showing the cell body, and the dendrites. And you can see right here, these are axosomatic synapses, not axodendritic. So you can see that, and it could happen either way. And again, going down, here's your axon hillock, here's your initial segment. So the summation is going to happen in between those two parts. Again, the trigger zone. And if you reach an action potential, and this multipolar neuron is myelinated, you'll have saltatory conduction from node to node over the myelin sheath to the nodes of Ranvier. And then again, as it's coming down, it's depolarizing and repolarizing segmentally through this cable unit. And then we'll reach the axon terminals, which is here. And it's actually showing that you have neurotransmitter being released. So the calcium channels had to be open due to that action potential. So this was excitatory in this case, okay, for the postsynaptic neuron, right? The postsynaptic neuron. So the presynaptic neuron, and this is a case of excitatory conduction, you have action potential, calcium channels open in the terminal, or the axon terminals, also known as botons. And then you have release of an excitatory neurotransmitter. So this is an excitatory neurotransmitter. And the neurotransmitter is the ligand. But of course, the neurotransmitter needs a receptor. The ligand has to bind to a receptor to open up the gated channels, the ligand gated channels. And the neurotransmitter is chemical, that's why they call that. Then you're going to have opening of sodium channels. Obviously, sodium's coming in, causing depolarization. The depolarization is an excitatory postsynaptic potential. And you have decremental conduction along the axon, each segment at a time. And then you have more opening at the initial segment of the axon, right? And then if it's excitatory, you have enough EPSP summated at the trigger zone, then the action potential will continue down that neuron, excitatory, and do whatever work it's doing to the next neuron or the next gland or the next muscle or the next neuron. Okay, so here's some neurotransmitters. It looks like we're going to talk about acetylcholine first because that's the most abundant neurotransmitter. And again, it's excitatory in the somatic nervous system. So it opens ion channels, but not always sodium channels, right? So sometimes acetylcholine can be inhibitory, but not in the somatic nervous system or somatic muscle contraction. In the autonomic the autonomic synapses, it can be inhibitory, especially with the parasympathetic nervous system. And I'll explain that to you. So in some cases, like always with muscle contraction, but in autonomic, it's different. In some cases, inhibitory, like in parasympathetic stimulation to certain receptors in certain parts of the body, certain smooth muscles, certain cardiac muscles, certain glands. And it depends on the organ. Really, it should be the target organ or the effector, because that's where the receptors are, the postsynaptic receptors, right? So excitatory in some areas of the CNS, like in the brain, right? And some autonomic, and in all somatic motor. So we're going to have to revisit this in a second to see what this is exactly talking about. And of course, it's inhibitory in some autonomic motor neurons. So this next thing is really confusing, so I'm going to take a second and just explain something. So acetylcholine is a chemical. It's a ligand. It's a neurotransmitter. So acetylcholine is released by many presynaptic axon terminals, right? So we have to go back and remember like the beginning of this nervous system business. You have the somatic nervous system, which is about muscle contraction, voluntary. And you have somatic senses that come in. somatic, which you cognate, right? Voluntary movement, cognitive sensory. And then you have the autonomic nervous system, which is not voluntary and not really cognitive either. So autonomic involves the parasympathetic and sympathetic nervous system, correct? So now we're talking autonomic, which is the two branches. Fight or flight is sympathetic, and parasympathetic is rest or and digest, or rest or repose. So this gets a little confusing, but think of it this way. This is the way you have to learn this, because the next slide is scaring me because I know it's hard to get through for years. So remember this. When you're talking about the autonomic nervous system and the somatic nervous system, think of the target, think of the effector. So simply, and now we're going to talk about motor, which is just efferent, coming from the CNS to the PNS. Yes, excellent. So everything is coming out either somatically or autonomically, motor, motor, motor. So with the somatic nervous system, those neurons are releasing acetylcholine, and they're always excitatory, and they're always making muscles contract by binding acetylcholine to those postsynaptic membranes. Always excitatory. And it's motor, right? It's motor. Now, autonomic, and it's always acetylcholine. Autonomic, now think of the effector. For the autonomic nervous system, there's three effectors. One is cardiac muscle. You know that pumpy thing in the middle of your mediastinum that's got cardiac muscle, completely involuntary, completely autonomic. The second effector is your smooth muscle, which is your blood vessel walls, your walls of your digestive tubes, walls of your respiratory tubes and reproductive tubes. So all muscle does is contract. So the third effector of the autonomic nervous system is glands, like sweat glands, like the adrenal gland, right? Glands. So now the neurotransmitter that's released to the effector of the parasympathetic nervous system, like the heart, the smooth muscle, and the glands, mostly heart and glands. For parasympathetic, that neurotransmitter that's released from the postganglionic fibers is always acetylcholine. So in the parasympathetic nervous system, acetylcholine is the neurotransmitter that's going to the effector, the target, that's going to do the work, like the heart beating or the digestive tubes contracting for peristalsis and moving food, chyme down the tubes, and reproductive as well, and respiratory. So acetylcholine is always released to the receptor. Now, sympathetic. Let's talk about sympathetic. Fight or flight. And I'm not going to bore you with me eating my burrito in the park today. Let's just talk straight up now. So the neurotransmitter that's released to the effectors 99% of the time, to the heart muscle, to the smooth muscle, to the glands, is norepinephrine, or noradrenaline if you like that, but it's a neurotransmitter. Okay? So that's really important. But I just mentioned the effector now. So in the autonomic nervous system, you're going to have preganglionic neurons and postganglionic neurons. Remember, a ganglia or ganglion is a group of cell bodies, synapses mostly, outside the CNS. So a ganglion is a synapse outside the CNS. So the preganglionic neuron comes from the CNS. You follow me? It's still PNS. It comes out. And then it has a synapse with a postganglionic neuron in the autonomic nervous system. And then that postganglionic neuron is going to go to the target, right? It's parasympathetic and sympathetic. The three targets are smooth muscle, cardiac muscle, and glands. So let's stay out of the autonomic, out of the somatic nervous system for a minute, because that's straightforward, acetylcholine all the way, right? So does that make sense? So in the autonomic nervous system, you have two neurons, preganglionic and postganglionic. They're both in the PNS. And the postganglionic is going to affect the effector, which is the target organ, smooth muscle, glands, and cardiac muscle. That's the best I could describe that. because it gets a little hairy.
And the neurotransmitter is norepinephrine.
For which part of the autonomic nervous system? See, this is where it gets difficult. You see, like for parasympathetic to the effector organ, the target is always going to be acetylcholine, always. And parasympathetic. For sympathetic, 99% of the time, it's norepinephrine for sympathetic fight or flight. So those three effectors. So I'm basically teaching the autonomic nervous system kind of early, but it's gonna be important later. Trust me, we can breeze right through it because you'll know it. Because you can't move fast through this. It's just it's a little bit too it's too much, really. Let's see if we can move on to the next page here. So now we're talking about the two types of acetylcholine receptors. Let's see if we have it here. No, it's not there. It jumped right to this. Okay, so know this. Let me write this out for you. Any receptor, and you have to memorize this in a way, any receptor for acetylcholine is called cholinergic. Right? And there's two types of cholinergic receptors. One is called nicotinic, and the other is called muscarinic. So you have to wrap it around your head. Whenever you hear nicotinic receptor or muscarinic, they're cholinergic and they're for acetylcholine. You have to remember that. This has nothing to do with epinephrine, a cholinergic receptor. And of course, you're going to have them on both. This is going to freak you out, but you're going to have cholinergic receptors on parasympathetic and sympathetic postsynaptic neurons. Because when I mentioned it before, when I stopped the slideshow, I was talking about release of a neurotransmitter to the effector, right? Which is tough to understand. So nicotinic receptors, you have to know, are for ACH, which is acetylcholine, and they're cholinergic. And this is the way they found them, by the stimulant called nicotine, right? The nicotine that you put in your vape or whatever. They're in cigarettes, right? Nicotine is excitatory. So that's how they found out about these receptors, right? So it is saying now that nicotinic receptors are found on the end plate of skeletal muscle because there's no real muscarinic. Muscarinic and nicotinic are both A and S. So just talking about muscle, it's at the motor end plate of skeletal muscle. In autonomic ganglia, these receptors are on the post-ganglionic neuron. So let me see if I can draw this out for you, right? So here's the CNS, right? CNS, I could spell CNS, right? Which is the brain and the spinal cord. And then we have a preganglionic neuron, which comes out to the PNS. Right, this is the preganglionic neuron, which has axon terminals, just like any other neuron, right? Axon terminal, and they're not heavily myelinated. It depends on what part of the nervous system, autonomic nervous system we're talking about. So now you have a postganglionic neuron, right, with an axon and axon terminals. Now this is autonomic. This is autonomic now, not just muscle, right? So right here, this connection is a ganglion. It's a synapse, of course, but this is called a ganglion. So this is the pre-synaptic neuron of the autonomic nervous system. And this is the post. I put synaptic, sorry, post-ganglion, pre-ganglionic, sorry. That's a better way to say it. Pre-ganglionic and post-ganglionic. Okay. So let's say this is the parasympathetic nervous system, right? So the preganglionic neuron is releasing what neurotransmitter? It's acetylcholine. So the postganglionic membrane has cholinergic receptors. So guess what? The postganglionic neuron in both the sympathetic and parasympathetic have cholinergic receptors, even in sympathetic. But if you go out to the effector, right, the heart, the blood vessels, and the glands, right, in the sympathetic nervous system, this is releasing norepinephrine. This is always acetylcholine. Always acetylcholine. This is always cholinergic receptors. But in the sympathetic, it's norepinephrine to the effectors. So that would be sympathetic. If it was parasympathetic, it would be acetylcholine going to these effectors, mostly the heart and the glands and the blood vessels as well. So acetylcholine is released to the effector in the parasympathetic nervous system, where norepinephrine is released to the effector in the sympathetic nervous system. But this ganglion between the preganglionic neuron and the postganglionic neuron is always cholinergic. Always acetylcholine. The first synapse always involves acetylcholine, both parasympathetic and sympathetic. In fact, these receptors right here in the postsynaptic neuron are nicotinic. So the first synapse between the preganglionic neuron and the postganglionic neuron involves cholinergic receptors for acetylcholine, and they're always nicotinic. So it doesn't matter in this synapse if it's parasympathetic or sympathetic, but out here to the effector, it matters. So all the receptors for ACH are cholinergic, and they're called muscarinic to the effector. And that's only parasympathetic. The norepinephrine receptors for sympathetic only are called adrenergic. This is adrenaline, right? So this is to the effector. So these could be different. They have receptors for both. You have cholinergic, muscarinic for acetylcholine, and you have adrenergic for norepinephrine. So adrenergic is for sympathetic. Cholinergic is mostly parasympathetic. There is one time where the sympathetic postganglionic neurons are releasing acetylcholine to the effector, and that's sweat glands. So that's one contradiction, if you will. So in the sympathetic nervous system, to innervate or activate sweat glands, the sympathetic postganglionic neurons are going to release acetylcholine, and they are muscarinic receptors because they're cholinergic. But just to the sweat glands, only to the sweat glands. So it's the only time that happens. Otherwise, for parasympathetic, the receptors on the effectors like heart, blood vessels, digestive tubes, all of that, which is smooth muscle, and the glands are cholinergic parasympathetic. And the ones that are on the target organs, effectors, are muscarinic. For sympathetic, everything except for sweat glands, all the effectors except for sweat glands have adrenergic receptors, heart muscle, all your digestive tubes, smooth muscle, blood vessels, respiratory, reproductive, and all the glands except sweat glands have adrenergic receptors released from the postganglionic neuron. So the ANS, the autonomic nervous system, you should know what that means now, have two neurons involved. And they're slightly different anatomically, you'll see. One's a preganglionic neuron, one's the postganglionic neuron. The first synapse between the pre and postganglionic neuron is always cholinergic. So acetylcholine is released from all preganglionic neurons in the autonomic nervous system. Can you dig that? Kind of, sort of. That's one of the most difficult topics in the nervous system, right? Those two neurotransmitters. Now, so acetylcholine is of course, the most abundant. So all preganglionic autonomic neurons all release acetylcholine. And the first synapse is generally called the nicotinic receptor, which nicotinic, you have to memorize, is cholinergic, therefore it's acetylcholine. So adrenergic is on the effectors. It's never in the synapse between the pre and post ganglionic neuron. It's always to the effector only, norepinephrine. Fight or flight, right? Fight or flight, acetylcholine. So let's see the comprehension on this. When am I supposed to get sick with this vaccination? I'm feeling pretty good. Drinking water like crazy. Am I going to be okay?
Pfizer? Did you have the Pfizer or the Moderna?
I had Pfizer.
Oh, then I think you're okay. The Moderna, I had the Moderna, and from the sample I've taken from people I've known, Moderna knocks you out the next
I
day.
kind of heard the same thing. I kind of
Yeah,
heard the same
Pfizer's
thing, yeah. Yeah, so I'm okay. I mean, I got to teach tomorrow and tonight, but it's okay. You know, what am I doing sitting here drinking water? So I should be fine. Don't worry about me. Don't call 911 yet. Okay, anyway, so let's think about this. Think of the effector. I'm going to give you the hardest question, see if you can figure this one out. Let's go back to the burrito. I'm eating my burrito, and the bear comes out. It's a bear today. It's a bear. So I go into sympathetic stimulation immediately. So all my preganglionic neurons are going to release acetylcholine to the postganglionic neuron. So now let's talk about the postganglionic neuron and what it receives and what receptors we have on the effectors, right? So my digestive tubes, my small intestine, we'll say, is in the walls. There's smooth muscle, which is supposed to contract to help my burrito moving in the right direction, right? Well, it's a chyme at this point. So I'm on the sympathetic stimulation. What neurotransmitter is released to my digestive tube smooth muscle? It's either epinephrine or norepinephrine or acetylcholine.
acetylcholine to stop.
Yeah, you think that. Right.
What?
It's norepinephrine. It's norepinephrine because I'm in to the effector, remember? To the effector. So now norepinephrine is going to bind to the adrenergic receptors on my digestive smooth muscle. But here's the question. Now think about this. I'll give you 5 seconds. Now, is that norepinephrine going to be excitatory, or is it going to be inhibitory towards muscle contraction of smooth muscle in my digestive tubes? Excitatory or inhibitory?
Inhibitory.
It's inhibitory, right? So norepinephrine is not always excitatory. It depends on what the target is, right? What about to my heart? Now, again, I'm the bear is there. 10 seconds, I have to decide. So now to my heart, what is the neurotransmitter being released to my heart muscle to contract or speed up?
Acetylcholine?
No, sympathetic to the effector. It's norepinephrine. And it's going to have beta-1 adrenergic receptors, beta receptors in the heart, beta-1 because it's only one heart. So in sympathetic, epinephrine is still released to the effector, but it's going to cause excitation, muscle contraction, harder, heart rate increased to get cardiac output to my brain so I could decide whether I'm going to finish my burrito, I'm going to fight the bear or run away from the bear. I think the answer is clear. I'm going to lay down and cry and hope for the best. But I have to make that decision quickly. So I have to get the blood to my brain as fast as possible. And my blood pressure will increase too, because there's adrenergic receptors on my blood vessels, which as I'm releasing norepinephrine to the blood vessels, they're going to cause my blood pressure to increase by contracting and causing vasoconstriction. Okay, so those were kind of easy questions, really. But you just had to think about it. No, the hard question would have been, the bear's chasing me, and I asked the question really fast, and I said, what neurotransmitter is my preganglionic sympathetic fibers releasing? And the answer to that is acetylcholine, because every preganglionic neuron releases acetylcholine into that ganglion to the postsynaptic membrane, postganglionic neuron, which has only cholinergic receptors. And what are cholinergic receptors for? Acetylcholine. Muscarinic are receptors for acetylcholine on the effector. So muscarinic, you have to remember, are cholinergic. It's always for acetylcholine. Adrenergic is always for norepinephrine or epinephrine. Adrenergic, right? We talked about beta blockers, I think, last time, and how you're blocking the binding of, well, technically not blocking the binding, you're blocking the cascade after. So you're blocking the beta receptor activity. So when epinephrine's released, it doesn't cause an effect because the receptor is blocked. So beta blocker blocks the beta adrenergic receptors. So your heart rate will slow down and your blood pressure will go down. Like if you, again, you could probably look into every grandma's medicine cabinet, and they take beta blockers mostly for blood pressure, or to lower the strength, decrease the contraction of your heart, so it's not so taxing on your body. So beta blockers are pretty widely prescribed to keep your heart rate down and keep your blood pressure down. Like somebody asked last time, Jessifat, maybe it was you, about performance people dancing and performing, taking beta blockers just to calm them down, right? It kind of makes sense. Of course, I'm not promoting that, right? Any drug you take for any misuse is a complete disaster, especially for something like that. But I see where it makes sense why somebody would want to do something like that. Well, you might as well take an anti-anxiety too while you're at it and really get hooked on it. But every medication has a side effect, even if it's used correctly. So again, take that for what it's worth. But it affects your autonomic nervous system, like especially a beta blocker is going to affect your sympathetic response to the effector, like your heart, your smooth muscle, or your glands. Now, the real tricky question would have been, as the bear's chasing me, what neurotransmitter is released to my sweat glands? You'd have to say acetylcholine, because that's the only time the sympathetic releases acetylcholine in the sympathetic response to the effector, and it's sweat glands. So technically, sweat glands in the sympathetic nervous system have cholinergic receptors to release sweat under sympathetic stimulation. It's a different kind of sweat, though, right? It's not the same sweat that you release when you're hot. It's a sweat you release when you're nervous. It's a different gland, right? There's different types of sweat glands. One is a more viscous, nervous sweat, and the other one is more protecting you from hypothermia, which you release water from that. So that they're technically two different sweat glands, and they change through puberty. There's differences in chemistry, and it's affected by hormones. So, but also the sympathetic and parasympathetic nervous system. Kind of makes sense. So, yes.
So I just wanted to clarify. So in the example with the burrito and the bear, the norepinephrine would be simultaneously acting as an inhibitory and excitatory neurotransmitter, depending on which effector it's at.
Well said. Did everyone hear that? That was really well said. Yeah, it depends on the effector. I'm really trying to drive this effector thing home, because that's the organ or gland or tissue that's doing the work to make the change, homeostatically. Yeah, for sure. That's very, very well said. And if you understand that, then the other part's just nuts and bolts, like the preganglionic, postganglionic stuff, what a cholinergic, muscarinic, nicotinic, and then adrenergic receptors. But and that's really important. That was a good synopsis. Yeah. All right. Any other questions?
So to the Pete, I was fan. As well said as it was, I still can't grasp my head around.
Yeah, no, I'm telling you, it's
the parasympathetic now. I'm sorry. So according to the examples, the parasympathetic gets norepinephrine at the effector in order to create an inhibitory response.
Sometimes excitatory though.
Sometimes, and sometimes excitatory because it's nicotinic, right? So it constricts
No, norepinephrine is not nicotinic. That's
not. It's muscarinic. Okay.
That's also cholinergic. That's the nuts and bolts you have to work out in your own brains, right? Because I don't know how to tell you to do that, to remember that, except showing you thousands of, but we're not studying receptors.
Norepinephrine is, I'm sorry, it's also cholinergic?
No, those are adrenergic.
Both are adrenergic.
Hold on, let me recap.
Okay.
Just the receptors, and this is everywhere, and even in somatic muscle contraction. For acetylcholine, you need a receptor, which you told me before very clearly. So, and the ligand is acetylcholine. So all receptors for acetylcholine are called cholinergic. It makes sense, right? The word cholinergic, right? So again, you have to remember that you just learned it. I'm amazed that Yaya even got that down in the first try. So the cholinergic are two types. There's nicotinic and there's muscarinic. Those are purely cholinergic. And that's what you have to remember. Do a cheat sheet, put it on your hand. I don't care. Take the test. I'm not going to be there anyway. You can look at your hand. What do I care? As long as you learn it. So you're learning it now, right? You're learning it now and you listen to the recording. So all cholinergic receptors are for acetylcholine. Two types of cholinergic receptors are nicotinic and muscarinic. So nicotinic are typically in the autonomic nervous system between the preganglionic and the postganglionic neuron, right? But in somatic muscle contraction, it's just the nicotinic one because it's a straight up, it's not autonomic, it's just lower motor neuron to the muscle. And that's always acetylcholine, like we talked about before. Yeah, so now separate that cholinergic and now talk about norepinephrine. And remember, epinephrine is the same thing as adrenaline chemically. So norepinephrine is slightly different than epinephrine, but there's a noradrenaline too. So it's all basically have the same receptors. That's the best way to put that. So it sounds like I'm naming 4 different things, like norepinephrine, noradrenaline, epinephrine, adrenaline, but they all respond to the same receptors, or they're all attached to the same receptors, and they're adrenergic. All right. I don't want to freak you out by using the word catecholamine. I'll do that later in this PowerPoint if I make it. If I make it or the time makes it, whatever happens first. So adrenergic are receptors for norepinephrine, pure and simple. Now, like Yaya said, you're going to have adrenergic and cholinergic receptors on all those 3 effectors of the autonomic nervous system. But in some organs or effectors, the neurotransmitter is going to be excitatory, and others it's going to be inhibitory. Like the heart's the best example. So again, back to the bear and sympathetic stimulation. Norepinephrine is going to target the heart. Postganglionic neuron is going to release norepinephrine to the SA node, which makes the heartbeat faster and contract stronger. So in that case, it's always excitatory. But then now the bear leaves, right? The bear leaves, and now I could finish my burrito, right? So now I have to go back to digesting as well. So now I'm back into parasympathetic stimulation. So the neurotransmitter that's released to the heart is going to be acetylcholine. And that's inhibitory to the SA node, slows heart rate down. The SA node is an electrical node in the right atrium that it has automatic action potentials actually to contract heart muscle. So now the acetylcholine's being released instead of norepinephrine to the heart, and my heart rate will eventually slow down. The only problem though with that description is because you have another chemical called, it's the same chemical, but it's released as a hormone. So the hormone goes into my blood, it's endocrine. So my heart might beat a little longer because of the hormone traveling in my blood. Well, the nervous system's very fast. So, but either way, acetylcholine is going to decrease the action potentials in my SA node and slow my heart rate down. Has no effect on contractility of the heart. Only epinephrine does that. And the blood vessels, systemic blood vessels, when I'm in sympathetic, there's alpha adrenergic receptors actually on the, and I said adrenergic, which means it must be for acetylcholine. Wrong. It's for epinephrine, norepinephrine. So if I am releasing norepinephrine to my smooth muscle in my blood vessels, they're going to constrict because contraction causes vasoconstriction. And that increases my peripheral vascular resistance and increases my blood pressure. Like you ever like press on a hose, you know, the water comes out faster when you decrease the size of the lumen. So it comes out faster. So that gets the blood moving a little faster to my brain, which again, I'm deciding whether I want to fight or run away from the bear. Because it's really all about getting blood to the brain faster through your cardiac output and your blood pressure, systemic blood pressure. So you got to go back and you got to put all those receptors together, like the cholinergic for acetylcholine, muscarinic, nicotinic. And adrenergic is only for norepinephrine or epinephrine or adrenaline or noradrenaline, whatever you want to call it. All right.
Okay.
Then you have to know alpha versus beta, but I'm not going to go too far into that. Yes.
I was just gonna ask, so in the PNS acetylcholine, you said would be in the case of after the bear leaves, would be inhibiting the heart, and then would it also, if you're continuing to eat, it's now also
Yes.
exciting the
Excitatory
digestive
towards my
system?
digestive system. The smooth muscle. Remember, it's smooth muscle is the actual effector. Yeah, that's exactly right. Be careful when you say PNS, though. I try not to do that because it sounds like peripheral versus parasympathetic. But you're right. And some books will say that, like they'll say like PNS is for parasympathetic. So I try to avoid saying that, just not to confuse anyone. But that's exactly right. Yeah. So they don't work together very well. They're like evil cousins, those two nervous systems. They really fight. The only time which you'll see in reproductive, they kind of work together nicely for about 4 seconds, and that's it. So it's nothing to do with the bear, I hope. Okay, let me just do 5 more minutes and we'll get out of here. Was that clear? That was really good. I mean, that's a tough topic for anyone, let alone the first time you're hearing this. I have grad students that wrestle with this for a while. By the way, this is going into the Whitney, this drawing. So if you want an autograph, let me know. Oh, muscarinic, right? And this was actually extracted not from cigarettes this time, or tobacco, but this is from mice, right? Or mushrooms. They did it within mice. They used this poisonous muscarine in the mice synapses. Okay, so these again are acetylcholine. We're still talking about acetylcholine here, by the way. We're way ahead of the game. But this is still going on with acetylcholine. So again, this is more about, what are these things? Cardiac muscle, glands, and smooth muscle. They're the effectors, right? So the muscarinic receptors are on the effectors. The nicotinic were between the preganglionic and postganglionic. In the ganglion, that whole synapse called the ganglion. But we're talking about effectors here. And here's your ANS. They're not even saying ANS. This is only ANS. And ANS now I could say, because there's nothing else, it's autonomic nervous system, and the 2 branches are sympathetic and parasympathetic. And here's your effectors. Smooth muscle, right? Smooth muscle is not just in your blood vessels, in your digestive tubes and reproductive, respiratory, right? So they have muscarinic receptors, which is only for acetylcholine. So it depends where we're talking about, right? Like Yaya said, if the receptor is on the smooth muscle of the digestive system, then acetylcholine is going to be excitatory at that muscarinic receptor. If it's on the heart, SA node, then it's going to be inhibitory towards the muscarinic receptor on the heart, which is the SA node. So this is really about autonomic system, and only autonomic, and it's all motor. We haven't really mentioned sensory autonomic. There isn't much to talk about, but so remember when we first described the nervous system and the branches, you saw that the motor nervous system, the motor part of the nervous system, is split up between somatic and autonomic. And sometimes they use enteric, which means the digestive tubes themselves, which I remember talking about that at the beginning. So agonist and antagonist, these are evil cousins, right? These two things here. So again, agonists work together, like nicotinic and muscarinic, or muscarine and nicotine, and they both affect, or both bind to, or both are ACH receptors for acetylcholine, right? But they found out through these excitatory chemicals that it mimics ACH, and it can affect the same receptor. So an antagonist is inhibiting the receptor, right? So atropine, right? Atropine is an antagonist for a muscarinic receptor. And curare is an antagonist for a nicotinic receptor. Kind of have to remember that. That's kind of cool. So what the heck is atropine and curare? So atropine, did you ever go to the eye doctor? I talked about this, didn't I? You go to the eye doctor and they dilate your pupils by relaxing the smooth muscle. So they put atropine drops and you walk around like you can't take the glare, right? Because your pupils are dilated. Because they're inhibiting the muscarinic receptors. Curare is like, when, if I'm in Central Park and some crazy guys shooting blow darts at me to try to anesthetize me, right? Like in the jungle where they do that for animals, they use curare because it was kind of a naturally found chemical back in who knows how long ago. So that inhibits the nicotinic receptors. Same outcome though, right? You're relaxing, you're totally not allowing muscle to contract by inhibiting the receptor for acetylcholine, right? So it's kind of like a paralyzing, but a flaccid paralyzing where you kind of just drop, right? Because you can't attach the acetylcholine to the receptor, right? Let me just get rid of this. Any questions? Any more questions about that? Makes sense. And we'll talk more about other